right. So let's look at this paper. So this is the paper I call the Andino paper online. And this is a really good introduction to quasi-species theory. And it's written in a way that most everybody would be able to get it. Most everybody with a kind of a science background. And so I think you're going to have an easier time with this than you would have with most other kinds of papers. It's meant to be accessible, and it brings a fascinating topic to us in really wonderful detail. All right, so let's go and look at the pictures. It's always the best way. So step one, RNA viruses exist as a quasi-species. So when you're infected, it starts out as just one virion maybe coming into a person. But then, even if that one virion comes in and it only has one sequence, within a couple rounds of replication, the virus will actually have made mutations. And then once it makes a mutation, it'll keep replicating, and it'll start to have that mutation plus some other mutations. And so the first round, you might start out like this and make one little mutation and a couple bigger mutations. Second round, from this mutation, you're actually going to keep mutating. And so you're going to keep moving outward like this. And as we go on, the virus is going to continue mutating through successive rounds and making more mutations. So that's just how viruses do it. But each time there's going to be a center point of the cloud. And so here in this, the center point is going to be pretty close to the original sequence that came in. And so this is going to be the average. These guys are going to have some mutations. They're going to be mostly the average, but with maybe 2 or 3 other mutations. These guys will be mostly the average, but with 2 different mutations. This will be mostly the average with a different couple of mutations. Same here. And the average of this cloud of viruses, that is the center of the quasi-species. And so the center doesn't necessarily move very much or very often. But the individuals that are in the quasi-species can be really wild. And the cool thing about a quasi-species is that it's a bunch of genetically related viruses that probably share a common ancestor not too long ago, but that will act cooperatively without ever realizing they're doing this. And together, because they're this cloud of viruses, they will be able to overcome immune systems, things like that. By working together, essentially, and without realizing they're actually working together. So, all right, let's go have a look at this. So, first of all, you got to have the idea of a fitness landscape. And so here's a very simple fitness landscape. So as we go up and sort of raise off the floor, this is going to be higher fitness, which means we have more children per infection, if you like. And over here, this sort of plane is sequence space. So as you make a mutation, so imagine this point 1 is some combination of 30,000 nucleotides that equals a coronavirus. And as we make mutations, you're going to shift a little bit, in some direction. And so we'll imagine that you shift a little bit here, and then where do you end up on the fitness curve? Boom, you end up right down there. Or if you shift a little bit this way, boom, you fall off a mountain. So if there's a virus and it's on a very steep fitness curve like this, very steep fitness landscape, it's kind of trapped. And anything that it does by way of mutation is probably going to make the virus a lot worse in a big hurry. Now, on the other side, you've got a relatively flat, broad virus. One like this, it's not particularly fit in any one state, but it's not particularly unfit. There's not a big difference between the fittest and the worst virus there. And so if something anywhere near the top of these little blobs is okay and can be replicated, then these guys have a lot of ways that they can move. So imagine you're an immune system. Which one do you want to be facing? You want to be facing virus number 1, because if you can figure out a solution and the virus tries to mutate to get away from the solution, it can't. And if you're trying to figure out a solution and the virus just makes a quasi-species of different individuals, all those other individuals that are not the center of the quasi-species are just terrible. And you don't really have to worry about those. That's great. Virus 2 is the opposite approach to that. This is a relatively flat quasi-species, or relatively flat fitness landscape would be a good way to put it. And in this case, the virus can mutate and doesn't really affect the virus much, but it can totally get around whatever drug you just put in or whatever immune, T cell recognition you just came up with or whatever antibody you just made. Now, these are actually based on real viruses. And so the real viruses are shown. Actually, no, they went into this, but that's what these are. So the one up here in red at the top on the pointy peak, that is something very much like a virus called vesicular stomatitis virus, VSV. And VSV is normally a virus of horses. And this virus has also been used as the base to make the Ebola virus vaccine, which is something that we talk about in the summer vaccine course. And so what we're showing here is that, yeah, if you start out with a population that comes in pretty close to the center, because that's probably the only one that can infect, and then you start to make that quasi-species, as you make them, this is what happens. They mutate a little bit, and you end up with a bunch of dead or very weak viruses, and you have your original population that are still pretty much at the top of their game. So this is your typical virus that is just trapped. It cannot get off this fitness peak. It may be a high peak. It may be able to do really well, but it can't really go anywhere. It's stuck there. Down here at the bottom, this is meant to simulate a virus like HIV. And so HIV is highly variable. HIV changes quickly and doesn't pay much of a cost when it does change. So imagine we start out here at the center, and as we spread out, as we pick up mutations, the same mutations that made us just dead in the VSV case at the top are actually just about as good as the starting virus in the HIV case. Good enough to at least continue the infection and move on to the next person potentially. So this whole idea is called not survival of the fittest, but survival of the flattest, based on a fitness landscape. Because if you're super fit, but you're on a super pointy fitness landscape, then it doesn't matter. You can't change. And so you're basically stuck up there and you're going to die out. But things that have a relatively flat fitness landscape and are able to move around on there without much of a cost of fitness, those things will do just fine. They'll do quite well. So that's why viruses like HIV persist in this world. They are very flat. Okay. Next concept in quasi-species, because quasi-species are so weird. This idea that you're getting infected with a cloud of viruses, or that one virus contains an entire, like the seed to make a cloud of viruses. Yeah, kind of nuts. What you see here is these are again genome copies. And I think this might be poliovirus, or it might be HIV. I don't know. Let's say it's HIV since we're talking about ApoBec. So you remember ApoBec 3G, its job was to mutate some of the nucleotides of HIV just at random so that when it went to infect another cell, it would find that it had a mutation and the mutation would break it. And so these are viruses that have been just not treated. And these are viruses that were treated with either ApoBec to make mutations, or you can put in chemicals that'll force mutations to happen. And over here, this is the number of mutations per genome. So normally, the virus is actually striving for somewhere around 2 to 3 mutations per genome. That is actually the optimum. That is what the virus is trying to do in this particular case of, let's say this is HIV. And so if you push it, so they figured out in this same paper, this is from a paper by Shane Crody, I figured out that anything more than about 4 mutations was actually the absolute limit. You could take up to 4 mutations with the virus he was studying, which was poliovirus. And when you got into 5 or 6 mutations, then the virus just did not work very well. It was basically dead. So he called this point between 4 and 5 mutations the error threshold. And what he found is that the virus actually replicates right up near the error threshold. It needs to be in the Goldilocks zone, pretty close to the maximum number of mutations it can make, because that way it can evolve the fastest, but still on this side of the error threshold, so if something goes a little bit wrong, it doesn't just slip over, because once it gets over here, once you get past 5 mutations, you are on average going to be dead, for this virus. So this is showing you that viruses make mutations on purpose. Or rather, they are fitter. There are many studies that show they are fitter when they are able to make a certain number of mutations. You can take viruses and you can make them make fewer mutations. You can just make their polymerases super careful, and it absolutely destroys the virus. The virus is not able to cause disease. It is not competitive. It does not do well. These viruses make mutations because by making mutations, one virus can actually build the entire quasi species in a very short amount of time, with all the different mutations that a quasi species can have, but still around the same center. And then they can go out and together they can conquer the host. It's like a little mutant army that kind of gangs up on you and beats you up, and yeah, there's just nothing you can do about it. So this is from one of those studies, and this is one that we're going to cover in detail, and we'll actually look at the paper where that this came from because this is important enough. So this has also got that error threshold for poliovirus, which is going to be between 4 and 5 mutations. 4 is right at the edge, and once you get to 5, most of these viruses are dead. It's not a direct, like alive with any 4 mutations, dead with any 5 mutations, but that's an average, and that's been worked out scientifically. So this G64S is a version of poliovirus that does not like to make mutations, and so you can see this thing makes very few mutations over here, whereas the wild type normal version of the virus makes an average of about 3 mutations, per genome. And this is going to make no mutations per genome on average. And so what's the difference between these things? Well, if you put this one into a mouse, the blue one, that doesn't make mistakes, the mouse gets over it and is fine. And if you put this white one into a mouse, it moves from one tissue to another and eventually kills the mouse because it's able to make mutations. So the idea is that no one virus is the best at anything. No one genome is the best at anything. There will be an optimum. And so with a disease like polio, it's going to come in via the fecal to oral route. So by eating something that's come out the back end of somebody else, unfortunately. And once it's in there, it's got to first survive the stomach. And so there'll be a particular combination of genes that'll be best for surviving the stomach. Once it gets through that, it's got to get into an intestinal cell. There'll be an optimum there. Once it's in the intestinal cell, it's got to get back out again. It's got to get to the bloodstream. Then it can go to muscles. Then it can go to the nerves that connect up to those muscles. Then it can follow those nerves up to the spinal cord. And then it can follow the spinal cord all the way up to the brain. And that's where we actually get poliomyelitis, and it actually becomes lethal. And there's no single kind of the virus that's actually best at doing all those things and evading the immune system and replicating quickly and putting itself together efficiently. But by making a quasi-species, you can make sure that for every situation, if you can get one virus into the next cell at the next level, it'll generate a brand new quasi-species, and together one of those is going to get into the next cell, and it's going to make the quasi-species again. Or you may have multiple viruses that are able to work together, both infecting a cell, one kind of carrying the other, as we'll see for a dengue virus in a little bit. This is the idea behind quasi-species theory, that the viruses can actually work together, and that by making a cloud of viruses, the cloud is stronger than if all the viruses were each individually fit. So you trade less fit individuals, but diversity. And with that, the virus actually wins. It's able to conquer our immune system. And when you have this sort of unity over here where the virus is all the same, then the virus just doesn't do well. Yeah. So that's perplexing, and that's different than what you probably would have heard of in terms of survival of the fittest. This is a thing called group evolution. Because if one virus is able to break down the immune system, let's say of a host, that's actually better for the entire group of viruses, the entire species inside of a person. And so they are actually both competing with each other for access to cells to replicate in, and cooperating with each other in that they can actually help each other out. And when one overcomes part of the body, that helps another one to be able to do the same thing. So I think that's the end of the pictures. Oh yeah, and so this is the super simple version of the Vignuzzi experiment that we're going to cover in the next paper. But it took the poliovirus that's not able to make many mutations, and they put it into mice, and the mice got the virus, and then they were fine. They cleared out the virus, and they were okay. And it took the virus that's able to make lots of quasi species, put that thing in, and yeah, even though it was the same center of the sequence in both cases, this virus was not able to actually kill the mice. And or this virus was able to kill the mice. This virus was not able to kill the mice up here. And so that's the idea. And yeah, I do urge you to have a look at this one. This is the essential one. This is the one you want to spend your time reading. And then this is the key to understanding the other quasi-species papers that we're going to be talking about.